This is a Pinnacle how-to video. This particular video is the basic principles of filtering the results table. This video will show in a step-by-step manner how to change the display from a screening and quantitation or an antibody screening and quantitation workflow for technical replicates of a particular set of raw files that have been placed into 3 different groups. The way that we've already processed the data, and this is from the antibody screening and quantitation workflow, there were 6 different raw files that were placed into 3 different groups, so 2 technical replicates per group. The group names are shown over here on the left or the right hand side. We have control, hydrogen peroxide, and high pH. We've already processed the data, and there's another video on this. The data, as we've shown it, has already been scored, and we have the heavy chain, the light chain, and we've also processed 200 of the most abundant unknown or unassigned peaks from our particular raw files. So the first thing that we would like to do is first of all show how the scoring is actually set up. And you can first of all drop in the raw files, set up an initial set of scoring, process the data, and then you can go back in and you can change the way that you're scoring. And in order to do that, we would first of all click on the redo, and we could click down here to redo scoring, and it opens up another window, and we can look at the scoring parameters. And there's quite a few settings that you can use. The lowest score would have a black X, and this just simply indicates that there's only some evidence on the MS1 level. There's no product ions, or there's no MS2 that could be associated to that particular precursor. It wasn't triggered. The second level is a red question mark. So it has, in our case, it has an MS2. It has matched fragment ions, at least 4 of them, that would be attributed to whatever the peptide sequence is from our knowledge base. The next level has an MS2. It has even more fragments that could be assigned, so we're assuming this is a better match. And you have to have a matched MS2 for a larger percentage, at least 80% of your raw files, for it to be scored in this next category. The 2 highest categories are the blue star and the green trophy. So in addition to having the MS2s, it would also have a very high matched MS1 dot product. That is, the isotopic distribution profile matches up its theoretical. It would have a percent CV less than 20%, and it would have a signal to noise greater than 10 for it to be considered as a blue star. That is, it has MS2, it has MS, and it has percent CV, so it's reproducibly measured across each of the technical replicates for our different groups. And then finally, we will also want to put a further distinction, and that is assigned some level of relevancy based on our individual groups. And we're basing this on fold change. That is, there's a difference in fold change greater than 1.5 between the groups. So that's how we've particularly set this. You can change these up as desired. Once you're done, you can hit done, and then you can click apply, and it would reapply to your scoring. We've already done this. So now if we look at this, again here we're in this particular table on the left hand side. We see that we have the particular rows at this point just showing the proteins. If we want to get into the peptides and just kind of go through what exactly or how the scoring looks, we can grab one of these first of all just to show that it's an X. If we click on it, we can see the chromatograms, and in this case there's no real evidence for that particular peptide. In this case, you can see that there's no blue triangles underneath showing that there's no MS2s, but the isotopic distribution and mass accuracy are or can be attributed to this particular peptide. And again, the black X's are simply there in the antibody screening and quantitation with the assumption that your samples are very pure or enriched for your targeted protein. For other with more complex matrices, you may not want to consider showing any of the black X's. But the black X's could be used to create a targeted inclusion list that could be then set on a secondary experiment. The next level up that we have are the red question marks. If we click on one of these, first of all, and let's just zoom in, And we can see that the isotopic distribution for these two charge dates match up very nicely. They do all have an MS2 underneath. If we click on one of these, we can see that it does match, but it doesn't have greater than the 8 or 6 or 8 fragment ions that are matched or attributed to this particular sequence. We can also see that it's not really changing its area values across our different groups. And again, there is a video on the groupings. Briefly, we've got 3 different groups, 2 technical replicates, and the order, so the first 2 area values would be attributed to control, the second 2 would be to the hydrogen peroxide, and the final 2 would be attributed to the high pH. Finally, if we start looking at a particular peptide that has a blue star, again, the isotopic distributions match up very nicely. It's got MS2s underneath. If we click on it, we can see that we have a very high number of fragment ions that are matched and attributed to our particular sequence. And lastly, we can see that we have some of their peptides that are colored or scored with the green trophies, and this indicates that they have an excellent MS2, they have an excellent MS, as well as the low variance, but there is a fold change between groups, shown here between the complementary isoform or forms of this particular peptide, where one is native and one is oxidized. We can see that the fold change for the hydrogen peroxide for the unmodified form is significantly lower, and then for the oxidized form for the hydrogen peroxide, it's significantly upregulated, as we would expect. So in addition to being able to filter or score our peptides based on a number of different criteria that are attributed to the protein sequences that we've provided for the heavy and the light chain, we've also in Pinnacle been able to look for unmodified or unassigned peaks as well, and they are also scored based on is it isotopic, is it consistently matched, as you know, as well as what's going on in terms of the area values across each of the raw files. And again, you have the blue stars and green trophies, and we're primarily interested in this case in the green trophies, and how is it actually related to the peptides that we've detected and measured for our specified sequences. So the goal that we want to do is to change the filter and not really focus on anything other than the green trophies. And in order to do this, we can come up, click on the redo, and now we want to redo the data filtering. And so again, we get a window, and it's asking us, how do we want to filter? And we're going to filter actually at the peptide level, and we want to get rid of the X's, get rid of the red triangle or red question marks, yellow triangles, and we're only going to keep just the high relevancy. Those that have MS2s, MS, and are changing levels, and when we hit apply, it will update our table. It takes very little time to do so. And so now if we click in and expand on the heavy chain, we can see only those particular peptides that have changed levels, and we can look across here and evaluate how they've particularly changed. And we can also do the same for, in this case, the unassigned peaks as well, and utilize this list to perhaps go in and evaluate it further, and exactly how is it changing levels, which raw files provide a difference in fold change, and we can utilize the precursors and evaluate this further in either a secondary experiment, or we can use it to specify and process the data. And this will end this particular help video.